Hey everyone, Lance Johnson here. My apologies for the delay in bringing you new podcast content, but I am back and I am super excited, not only for this episode, but the next few episodes that I have coming up for you. In this episode, you're going to get to know me a lot better. Fun fact, on August 10th, I turned 30 years old. The 30th birthday is a major milestone of a birthday, right? On my birthday, my mom sent me a school paper that she had saved from when I was in 6th grade. The paper was titled, When I Turned 30. In that school paper that I wrote when I was about 10 or 11, I wrote about what I imagined my life would be like when I was 30. And what I had written was hilariously different from my current life. I'm going to read what I wrote for you, but first, for those who don't really know me, let me refresh you on a few things about myself to help you understand the differences. I am a freelance video editor who lives in Brooklyn, New York. I rent an apartment with one of my best friends. I have a dog named Ruby, and my hobbies are creative based. Take this podcast, for example. All right, that's enough information. So here's what young Lance wrote, grammatical errors and all. Though I will note that you can't hear the font that I wrote this in. And I would have to say this one probably reminds me the most of like the font that was used in Rocko's Modern Life as a kid or something like that. Anyways, here's what I wrote. I will be a doctor. I will live in a 2-story house that is located in Georgia. Inside my house, it will look very clean, with Picasso paintings on the walls and chairs to sit on. It will look different from my parents' house because it will have swimming pool and you'll be able to swim anytime you want. I will probably work at doctor's office because I already know I have a talent in doctoring. My pay is around $135,000. I get to work at 9 a.m. and get back at 8. I like to relax in my free time and take vacations in the mountains. There have been so many inventions by then that there is no more beds in the world, and we don't have to worry about cavities anymore. The hardest thing about my life will be having a mistake in a surgery, but new tools makes it easier to finish better. All right, that's what I wrote. Let's recap how my life is different. For one, I'm not a doctor, and while I don't make $135,000 a year, at least I don't have to worry about making mistakes during surgeries. With that being said, out of everything I wrote, I think the main thing that made me laugh was the not having to worry about cavities line. Due to my candy intake as a kid, I had so many cavities. And to be honest, I have a root canal coming up, so not much progress there. I did have a few things happen though. I pulled off having chairs to sit on in my apartment. That's pretty cool. So what's the point of me reading that? For one, I think it's funny. And I thought I could give a little more insight to myself since I am the host of this podcast. Two, it's proof of something that I want to talk about this episode. It's proof of how clueless we are about what the future has in store for us. Yeah, maybe now with a slightly smarter brain, I could better predict what my life would be like in 20 years. But my point is, you can't really predict the future. For example, at the time I wrote that, I didn't know that I had the eye disease retinitis pigmentosa, or RP for short. Like the future, RP is something that is not predictable. I have no clue what my field of vision will be like when I'm 50. I read somewhere that on average, most people with RP are considered legally blind by the age of 40. That's a tough pill to swallow. I guess this is where hope comes into play. And while there currently isn't a cure or a treatment for RP, In 20 years, when I am 50, will there be one? Will it restore my vision, or will it just allow me to keep the vision that I have at that point in time? Will it be a surgery that I'll have? Will it be a device that I wear? Who knows? Also, when I'm 50, I may have children. As someone with RP, will my children inherit it? And most importantly, will I have a swimming pool at my house? Well, besides the pool question, I'm looking to have some of these questions answered. So that's my cue to bring Kristy Lee on the podcast. Kristy is a certified genetics counselor, and she happens to be my personal genetics counselor. Kristy and I had a great conversation where we discussed genetic testing, gene therapy, the types of genes involved in eye diseases. We even go over and compare the genetic test results from blood work of not only myself, but my mom and sister, who also have RP. But most importantly, we talk about the steps to take if you are interested in completing genetic testing to better help you understand your specific disease and potentially help find a cure for it. Let's jump into that conversation. Hey, Kristy, how's it going?
Hi, Lance. How are you?
Good, good. Thanks for coming on the podcast.
Yeah, no problem.
You're in Chapel Hill, North Carolina, right?
Correct. I work for the University of North Carolina at Chapel Hill for the Department of Genetics.
Nice. And you're a genetics counselor there?
That's correct.
More specifically, you're my genetics counselor.
I am.
I feel a little lost when it comes to gene therapy, and I that's why I brought you on. So I think I could use the education, and maybe my listeners might enjoy it too. So I guess first off, can you just say what is a genetics counselor?
Oh, genetic counselors are considered allied health professionals. We are trained specifically in genetics and genetic conditions. We are trained to assess risk within families that have certain conditions in their family, or sometimes in the prenatal setting to look at all sorts of different types of risk. We are trained to not only help determine risk and teach families what that risk is and what that means for future children, but we also try to be that resource to explain what the condition is, what are other health issues that might go along with that genetic condition, and then point people in the direction of resources to best cope with their genetic condition, and or link out to other individuals who might help create social communities and so forth. So we're kind of teachers and resources.
Awesome. So in a traditional sense, when I think of the word counselor, mm-hmm. I imagine that I go into someone's office, they ask me questions about myself, and then they try to give me advice on how to better myself or set me up for success in the future. Would you say your role is basically that on steroids?
I haven't thought of it that way, but I suppose so. You know, it's not your typical counseling situation where we're going to sit and talk about your feelings for, an hour and a half. We certainly do that, but we try to do that in ways that make folks comfortable. Like if they need to talk about those things, that's what we talk about. If they need to talk more about the science, that's what we talk about. So we try to figure out sort of what folks need to get out of a visit and encounter with us and how we can best help. So it's a little different than a traditional counseling session that you would have with a psychotherapist or a psychologist. But we do indeed have elements that makes sense to call it a genetic counseling session.
Definitely. I remember whenever I first was introduced to you, and I was told your job title, genetics counselor, and that's what I thought it was. Because, I go to the Kidner Eye Center, and I would do a bunch of tests, and the tests can be kind of demoralizing, to be honest with you.
Yeah.
The field vision test and a lot of photography of the inside of the eye, just a lot of just kind of monitoring of your vision. Nothing's changed. There's no cures out there or anything like that. So I thought your role was to kind of make me feel better about the whole situation, which, to be honest with you, I had mixed feelings on. I didn't know if I wanted to talk about it. But anyways, I'm glad I did. With that being said, I did give blood because, as a genetics counselor, you need to have some data and some things to look at and to chat with me about. Mm-hmm. And on top of that, my sister and mom both also gave blood for genetic testing. So we uncovered some information from that, and we won't get into that right now. So for the listeners out there, that's coming up. But I was thinking, before we dive into my specific genetics when it comes to my eye disease, which is retinitis pigmentosa, you could give the listeners a quick 101 on genes and genetics and what you're looking for.
Yeah, sure. So you can kind of think of genes as like an instruction manual to make a protein or something that the body uses in its everyday cycle. So what I tell people is, to think about having a set of instructions that aren't written correctly. And if that's the case, you can't follow those directions correctly. And it's the same thing with the body. If there's a gene that's not spelled right as a simplistic term, or has a gene change or gene variant in there, where it causes the gene not to be able to be read by the body, a lot of times it makes a protein that's very abnormal that can't function properly, or it may not make a protein at all. And so the body's sort of missing that important protein that it would otherwise have. And depending on, where that protein is supposed to function, be it the eyes or the heart or the kidneys, or sometimes in multiple areas of the body are affected, the gene will be dependent on sort of where those complications are going to arise in the body, whatever that gene is supposed to, what we call code for. So for eye diseases, what we're looking for, basically, genes that are involved somehow in either the way the eye is built or in the way the eye functions, or the cells within the eye function. So there are some cells that are important for getting nutrients to parts of the retina. If those cells aren't working because of a particular protein, then the cells don't get nutrients and they die. So that's just one example. But what we try to do is figure out which of those many, 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 many genes isn't working well. And I tell people it's kind of like when you have a car that isn't running. Until you know what part of the car isn't running or why it isn't running, it's really hard to do anything about it, right? So it's kind of like that first step to try to figure out, okay, what's wrong? Which gene is wrong and not working correctly? And then we can focus from there. And from there, we should be able to tell families how it's running through their family. So if they wanted children, what's the chance they're going to pass it on? Or even if they don't want children, what are the other family members that might be at risk of having the same condition? So that's sort of where it goes. So we get a blood sample. We can also do saliva these days. And we take a list of genes. We call them a panel. And the laboratory will look at all of the genes on that panel. And the gene panels now are very large. Some of them are close to like 500 genes. And so the lab will read the genes kind of like a book, making sure that there aren't any typos, if you will, or places where there should be a G where there's a letter T or something like that. If you remember, the genetic alphabet has 4 letters, A, G, T, and C, just like our alphabet has 26. And so the gene will read something like A, G, G, T, T, C, T, T, A, G. I'm making that up, of course. But the lab is trying to make sure that all the A's, G's, T's, and C's are in the correct order. And if there aren't any that are correct, or if there are any that are incorrect, then we get a report that says which genes have what we call gene variants that may or may not be a potential cause.
Very interesting. How has this study evolved over time? Like, what did genetic testing look like 10 years ago?
Okay, so genetic testing, when I first started, which was like 13 years ago, was archaic. We basically had a handful of genes that we could test for. And we didn't have these large tests where we could look at 500 genes at once. We actually had to pick out which genes that we wanted to look at. So we didn't know as many genes as we do now that can cause eye disease, number one. Number two, we did not have the technology to look at mass numbers of them. So we had to be really specific. And so people were getting testing for, just a handful of genes. And we know that probably well over 100, 150 cause retinitis pigmentosa all by itself. So it really was incomplete testing, and it was expensive. Now testing costs about the same thing as it did 13 years ago, and we can look at, 300, 500 genes with one test. So it's really changed, night and day. And it's allowed more people to get an answer for why they have their eye disease, which has been really great.
Yeah, and that's definitely something I am interested in. And I assume my listeners, if they have eye diseases or anything else, giving blood to get to see if you can pinpoint that would be probably a point of interest for a lot of people.
Yeah, it is getting even more popular with the idea of gene therapy. So absolutely.
So when you say gene therapy, are you meaning that you can take that information from the testing and actually fix certain genetic, would it be a mutation?
The idea with gene therapy, I think in its most basic form, is giving that individual back a healthy copy of the gene. So we've identified on a genetic test that a particular gene isn't working well. The next step is, it's kind of like the car. Okay, now we know what's not working well. Well, let's fix it, right? So unfortunately, that's really difficult to do, but we are making strides to do that. We actually have one gene that we do have gene therapy for, which is great. It's called RPE65. Unfortunately, most people don't have that form of eye disease, but for those few that do, they actually have a treatment. And in those individuals who would benefit from that treatment, they have 2 abnormal copies of this gene. So we all should have 2 copies. But these individuals that would benefit from gene therapy, neither one of them works. So essentially what we're doing is giving them back a copy of the gene that does work. And what's been found, or the hope, is that we stop the progression of their eye disease, because without treatment, they would continue to lose more and more vision. The reality of it is that seems to work so far, but it actually may improve vision a bit, which would be outstanding.
Interesting. So what you're saying is, in some instances, you can pinpoint the specific gene that was written incorrectly, and you can go in and alter it and correct it, and sometimes even reverse the eye disease?
Yeah, that's the hope. A lot of this work is still being done in clinical trials right now. And even before then, in sort of the animal model setting. But the hope is that work that's being done in animal models will give us the information and data we need to learn from in order to get to the clinical trials where we can actually study it in people, that will eventually give us enough data to get FDA approval, like the one gene RPE65 has been able to accomplish. So that's the goal, but it takes a lot of research and so forth to sort of get to that. Yeah.
I have a silly question. It's because I have no clue how it would work. And I'm sure other people are probably thinking the same thing. How do they change the genes? Like, how do they go in and alter that?
Right. So one of the ways they sort of have to figure out a way to package the new gene up. This is for the recessive forms right now. Mm-hmm. So the RPE65, we'll start there. So for that particular gene, what they had to do is they had to figure out how to package it so that we could deliver it to the eye. Okay. And it turns out that the eye is a phenomenal place to do gene therapy because it's kind of a closed system, if you will, and it's somewhat easy to get to, right? You don't have to have major surgery or anything like that. So this sounds kind of scary, but they actually inject the treatment into the back of the retina with a needle. And the medication that they're giving has been made using viruses that have had their DNA taken out. So they don't act as viruses anymore. We use them as kind of a vehicle.
That's insane.
Yeah, isn't that crazy?
I was not expecting you to say that you would inject with a needle viruses into the eye, and that.
Yeah. So they've been transformed. So we take the viral DNA out, and then we put in, and by we I mean scientists, put in the packaged new gene that does work. Along with some other DNA to help it get inserted into the our genome once it gets in. So it gets injected, and it doesn't get injected willy-nilly. So the doctors use the eye test that they've put you through to figure out sort of where are places in your eye that have living cells still, because you don't want to give this treatment to a portion of the eye where you've had too much cell death, because we need living cells to take in this medication and let it incorporate into those cells, sets of DNA. Mm-hmm. So that's how it's being delivered at this point. And so for the recessive diseases, again, the goal is to give the body back a copy of the gene that it can read and make a protein from, right? But for the dominant forms, we might have to do a little bit more work. It's more than just giving a copy of the gene. In some cases, it's going to be shutting down the abnormal copy. And so you and maybe some of your listeners have heard of CRISPR-Cas9 technologies that have been in the lay public. For years, Lance, people would sit in clinic with me and they would say, isn't there some way you could just, I don't know, like white out or just like clip out the bad part and just replace it? And, we used to laugh together and say, it's kind of science fiction. But it turns out that science fiction is now in the research lab. They are actually trying to figure out sort of how to cut out the abnormal variant or mutation, as it's sometimes called, and then replace it with the correct message, or even just trying to shut down the gene that's not making a normal protein. They're able to actually do that in the laboratory. So those are the types of things they're working on, and if they consistently can do that and be successful in animals, then they can move it up to human trials.
That's wild. My mind is still struggling to wrap itself around that. Yeah, it does sound like science fiction. What I imagine is like in the future, you can plug yourself into a computer and then your genes pop up, your genetic code pops up, and then you just like can change it yourself. That seems like it would be pretty easy.
Scary.
Yeah, a lot of downfalls to that too, I'm sure.
Yeah.
You wouldn't want the wrong person to plug you into that computer.
Exactly. It's funny. Probably important to point out to you and your listeners also that doesn't change the genes in the rest of the body. So if you went to have children, your chance to have a child with retinitis pigmentosa or whatever you've had gene therapy for is going to be the same as it was prior. So really, you're just treating the individual themselves.
Cool. Yeah. All right. So yeah, we'll get more into that in a bit once I think we go over the results of my blood work as well as my mother and sister. So, but one thing before we get into that, because I'm curious about this gene therapy is what's the difference between gene therapy and stem cell therapy?
Oh, yeah. So, good question. So gene therapy, we've kind of talked about, we're trying to give a gene back to the body that works or correct a gene. Stem cell therapy is giving back the eye cells that are healthy and functional. So the idea is that we can actually take now your own blood cells and actually turn them back into what we call stem cells, which were what your cells were before they turned into a blood cell or a heart cell or a lung cell or a brain cell. They all start as stem cells. So we go back to the beginning, make them go back to just being cells, and then we can make retina cells out of those cells. And then we can take the retina cells and give them back to the back of the eye. And hopefully, being successful there, they should have, be free of the damage that the cells that you were born with would have, and hopefully replace some of the cells that had died from the disease. So that doesn't change anything about the health of the genetic information. That would still be the same as whatever you were born with, but it would give the opportunity to create retina cells again and use those. So there are some studies that are using the individual's personal cells, and there are some studies that are using someone else's stem cells. The reason for using someone else's stem cells is because, say, if we did stem cell therapy on you, those cells are going to have the same fate that the ones you were born with have, right?
Okay.
Because they're going to have that same genetic defect, right? So they're still trying to figure out, what are the pros and cons of using your own stem cells versus someone else's stem cells. One of the cons is immunity, right? So if we start giving you someone else's retina cells, your body might react to that, right? So that was the idea of using your own, but then you either are going to have to potentially have stem cell therapy on multiple occasions, or we've got to figure out a way in the future to maybe combine the two therapies. I mean, that would be ideal, but again, is probably years away, is being able to take the cells, the stem cells, correct the genetic defect, and then grow retina cells from there.
So when you said taking someone else's stem cells and putting them into my eye, there might be a negative reaction to that. What would exactly happen?
Well, the worry is that your body will reject those cells, and the benefit from having, new retina cells will have been lost because your body will react to them as sort of a foreign invader, kind of like a virus, and it's going to go and destroy those cells. So that is the fear. So the question is that, do we try to combat that by putting you on immunosuppressants, which have its own kind of complication because you suppress the immune system in order to allow the stem cells to be able to grow and work, versus, maybe the idea of having to do multiple series of rounds of stem cell therapy. So those are all things that we're still trying to sort out.
Man, that's wild stuff again. All this is like incredible.
Yeah, it's cool stuff.
It's simultaneously hopeful and terrifying.
Yeah, absolutely.
So, yeah, I think this is a good segue. I wanted to talk to you about my blood work. I gave blood. I want to say this is maybe in 2017, I believe.
Yeah.
2017, I gave blood for genetic testing. And then shortly after, my mom and sister both did as well, with the goal being we can kind of analyze the three of us and kind of pinpoint commonalities and kind of maybe discover some information, maybe pinpoint specific genes that kind of caused the RP we all have. Since I'm assuming I inherited it from my mother, so did my sister, but that's all I know. So I guess the point of the testing was to maybe get more deeper in the weeds and figure out the deep details there. Should we go over my results first? Maybe I should just let you kind of.
Yeah, I can integrate.
Okay, so what we're going to do is Kristy has the results of myself, my sister, and my mom, and she's about to kind of dissect them and tell me some information that I haven't even been told yet. So this should be interesting.
Great. Okay. So when we tested you back in 2017, we used a lab that would allow us to look at several genes at once. And I think we looked at probably close to 130 genes in you at that point. And the laboratory that we used allowed us to send a sample from your mom as comparison. So based on your family history that we chatted about when we talked in clinic, it looked like that your family had a dominant form of RP. And the reason for that is because, as you mentioned, your mom has RP and you have RP and your sister has RP. Well, we can't say for sure that yours is dominant based on your family history, but that's what it looks like. That's what the family history is suggesting. And so we were basically, the lab was looking particularly for gene changes or gene variants that you and your mom shared. It's a really neat way to do this because you and your mom should share on average about 50% of your genes, right? She gave you one of each of her chromosomes, which have genes on them, and you got the other half from your biological father, right? So your biological mother gives you half, your biological father gives you half. And so you should share about 50% of your gene variants with your mom. And what we can take from having your mom sample is we can basically eliminate gene variants that you and your mom don't share, okay? Because we would suspect that, you inherited this from your mom, just like you said. And any gene changes that you didn't get from your mom probably came from your dad. And since he doesn't have a family history of this, we can kind of set those aside. Having tested your sister later on, we were able to whittle down that result even further. So when we tested you and your mom, there were 2 gene changes in genes that can cause retinitis pigmentosa when they're not working well that you and your mom both had a gene change in. So there were 2 of them.
Okay.
When we compare them to what your sister's test results show, a year or so later, your sister only has one of those 2. And again, since you all have RP, we would expect it to be the same type. And so it should have the same gene that's not working well. And so we would expect that any potential answer is one that all 3 of you have. And that narrows it down from 2 now to 1. So now you can see how important family members can be when there is a family history. There's not always a family history, but in your family there was, and so testing multiple members was helpful. So the gene that's sort of left for us to think about is called SNRNP200. I like to call it Snurpin 200 for short, but that's my nickname for it. But that And that gene, the lab has found a change in you and your mom, and a different laboratory found it in your sister. We can't say for sure that it's abnormal and harmful to the gene. But what we can say is that it does cause RP when it doesn't work well. It is inherited in that dominant fashion, and it does look suspicious. So that might be the answer for your family. Like most genetic changes, a lot of them are real private to one family, so we can't compare other families to your family. So we don't have another family that's got this exact same gene change that we can say, okay, both these families have retinitis pigmentosa. Now we've got an answer. But, over time, I'm hopeful that we'll be able to figure this out and say for sure one day whether or not this is the gene that's causing RP in your family. Right now, this is the most suspicious looking gene.
I think just for my sake, I'm just going to just bank on that being the gene. It makes the most sense to me. Basically, I just kind of want to summarize what you said just to confirm that is what is being said. We did blood work. Out of the three of us, there was one commonality. So we can kind of keep an eye on that specific gene. And there are no current gene therapies for that specific gene. But since now we have the knowledge of that gene potentially being the reason for my family's RP, if one arises, it just helps it be on your radar. Is what you're saying?
Yeah, absolutely. So yeah, like we talked about, there's really only one gene therapy right now. There's some on the heels of that one. So definitely there will be more, but for your particular gene of interest, there's not a treatment or clinical trial right now, but absolutely that could change. And so we want you and your family to know when there might be opportunities to think about clinical trials. And it doesn't mean that, you would automatically sign up for one. It just means, you'll be in the know. And at least if you don't want to participate, you'll know they're working on your gene, which is kind of cool too.
Yeah, I think it's great information to have. Is there any other information we could uncover from that? Like, how we were suspicious of it being dominant. Is that information? Are we able to uncover that type of info?
From this standpoint, I think we're still thinking that this is dominant, and certainly this gene would fit that pattern of inheritance. So, at this point, I would still tell you and your sister that if you had children in the future, that they would each have a 50-50 chance of inheriting this condition from you all. Whether or not this is your gene or not, that's still what we would say based on the family history.
So I have a 50-50, just based off of the data we have, it's likely that there's a 50% chance that my offspring would have RP.
Correct.
That makes sense because, I have two siblings. My sister has RP, but my older brother does not. So I guess he was that 50-50. That other 50%.
Exactly. Yeah. Well, most likely there are some dominant forms where you can inherit the gene change and you actually don't have symptoms. And that is a very complicated mystery that we are still trying to figure out. It may be that there's some other genes that also play a role into whether or not people have symptoms or not. But more likely than not, it's probably that he did not inherit the gene change that has caused RP for you, your mom, and your sister.
So does that mean that gene is no longer present? So his offspring would not have that same 50% chance?
Great question. So if indeed he did not inherit the gene change that causes RP in your family, his kids would not be at risk. This is not something that skips generations on the genetic level. Actually, nothing really skips generations. Certainly traits can hide in certain individuals, just like we talked about. But if you didn't inherit it yourself, you can't pass it on. So if you didn't inherit something that's in your family, it's not going to not be in your genes and then show up again in your children's. So most likely his children would not be at an increased risk for retinitis pigmentosa.
All right. So that's good to know. I think my brother will be happy to hear that.
Yes.
Because he has 3 kids. So, with that being said, 50% chance, that's still pretty good odds. And then honestly, and if my children have RP, the goal is with things like gene therapy and all the research being done, that it'll be far more advanced during their time on Earth, and they'll have better advantages in terms of potentially correcting it. So that's the kind of whole point. While there may not be something for me currently, there may be 20 years down the road. And if I pass RP down through my children, then there might be something for them. So, the whole point is it's hard to judge. It's hard to predict, just like the future is. But these kind of tests can be helpful, and they're making progress. And I personally am glad that I did the blood work to figure that gene out, even though there's not specific therapies for that specific gene. So, like, with that being said, if someone else, say someone out there listening has not done genetic testing, mm-hmm. how would they get that process going if they wanted to kind of to see if they can learn any new information about what they're dealing with?
Yeah, that's a great question too. In the past, genetic testing has been difficult for some people to have because it's expensive testing, and insurance companies, sometimes don't want to cover genetic tests where we're not going to change how you're taken care of or your medical management, as they say. However, right now there are 2 opportunities for folks that are interested where free genetic testing is available for eye disease. They're both done by very reputable labs that do great work. And so there's no time like the present to actually try genetic testing if interested. Mm-hmm. So there are a few ways to go about this. For anyone who's interested in sort of finding a genetic counselor, there is a website called the National Society of Genetic Counselors where they actually have a find a genetic counselor search engine on there, and you could Google find a genetic counselor. I'm pretty sure that would be the top hit on Google. However, most counselors don't specialize in eye disease, so it might be a little difficult to get to a genetic counselor who specializes in eye diseases, although you might find someone nearby who can find a specialist. But there's one of the laboratories who is offering testing where they will actually connect you with a genetic counselor as part of the test. You'd have that option to talk with a genetic counselor via telemedicine. And so there are, even more opportunities than ever to get out that, to get to that opportunity. Some of the bigger eye centers, like Kittner Eye Center that you talked about at UNC, around the nation actually have a genetic counselor that works with the clinic. So if you are in a major city and there is a genetic counselor available to you through your eye clinic, that probably would be the easiest. If not, it might be worth a conversation with your retina specialist or ophthalmologist and ask them to get in touch with one of the programs that's offering free genetic testing. So one of them is the My Retina Tracker program through the Foundation Fighting Blindness. The other is a program called ID Your IRD for inherited retina disease, and that's offered by Spark Therapeutics. So those are two programs that are free. You would have to work through a doctor or a genetic counselor to access those, but those would be the best ways to access either a genetic counselor or work through your retina specialist or eye doctor.
Okay. So basically, you listed a few website resources. I'll put those in the show description. So if you're listening and you want to check those sites out to learn more, check out the show description. And say, this whole podcast episode, we talked about my results, which are very specific to the eye. For the listeners out there who have a different type of disability, is there any sort of gene therapies that can affect other parts of the body?
Yeah, actually, I also mentioned I specialize in hemophilia, so there are now gene therapies that are being investigated for hemophilia A and hemophilia B, which are very similar bleeding disorders. There's also another approved gene therapy for a rare muscle disease that's present from birth called spinal muscular atrophy that is now available. And there are also some other gene therapies that are being worked on for some metabolic disorders that affect how the buildup of storage waste in cells it causes disease in those individuals, and they're working on gene therapy for those folks. Duchenne muscular dystrophy, they've been working on some gene therapy for that. That's not quite ready for prime time yet. Trying to think of some other examples. They're working on cystic fibrosis, which maybe many folks have heard about. There's a childhood lung disease. Can also affect the pancreas. So yeah, they are working on lots of different types of genetic diseases that affect, hundreds of thousands of people.
Yeah. So I think that's a great segue into my next question. Do you have a specific organization that you want to give a shout out to that's doing a lot to kind of pave the way for this type of research when it comes to gene therapy?
Yeah, I think you and your listeners have talked about the Foundation Fighting Blindness before, and I will just say they're an amazing organization that has funded a lot of the early studies that have made the more recent current studies possible. But one of the companies that's kind of come to fruition because of the work they did in the research lab for gene therapy is called Spark Therapeutics. And they actually benefited very much so from the early work that the Foundation Fighting Blindness funded. But Spark Therapeutics was developed in 2013 out of Children's Hospital of Philadelphia, who has been years doing work in gene therapy. It's kind of been a center to look upon for their work there. And a group of researchers from Children's Hospital of Philadelphia, we call them CHOP for short, came together and created Spark Therapeutics. This is the company that has made the gene therapy that brought about the one FDA-approved gene therapy that we have for retinal disease, that RPE65. So they're the ones that ran the clinical trials in order to bring this to the FDA and get approved. And they are working on a number of other retinal diseases as well, in addition to, the RPE65. So they have other diseases in clinical trials for eye diseases, but also other types of diseases as well. So they're a company that's also sponsored the ID or IRD genetic testing program. What they're trying to do is get, I guess you would say, a catalog of folks that could potentially qualify and might be interested in future clinical trials by offering this genetic testing is a free service, you agree to let them keep your name in a database and potentially contact you should you qualify for a trial they're running in the future. So for folks comfortable with that, it's kind of a nice way to get your genetic testing done on their dime, but also be kept in the loop about future trials that you're not having to participate in because you're not obliged to, I should say, but could be alerted about that. But anyways, they are doing great work with retinal gene therapy trials as well as other types of gene diseases. And I'm very excited about the work they do, and I hope they continue to have a lot of success doing it.
Awesome. Yeah, that sounds great. So Spark Therapeutics, I will list the link to their website if you are listening and you want to learn more about them. I appreciate that info. I was aware of Spark Therapeutics, but I feel like maybe I need to do a deeper dive now into learning more about them. So thanks for bringing that up.
Sure.
But yeah, Kristy, I think we covered everything. I feel like it might have been a selfish episode for me because it's information pertaining specifically to me. But hopefully, it's helpful to someone out there listening who maybe just wanted to learn a little bit more about gene therapy, or maybe there's someone out there who has that gene that you listed. And thanks for your time. And I had a great time talking with you, and I plan to keep in touch and keep you in the loop on what's going on with me and my family. Fantastic. So take care, Kristy, and chat soon.
All right. Thank you for the opportunity, Lance.
All right. Bye. Well, that does it for this episode of the See Through podcast. I'd like to give another shout out to Kristy Lee for coming on and chatting with me. It was a pleasure. I learned a lot. A lot of it was over my head, but I think I'm a tad bit smarter after this. Kristy listed out a lot of different ways to get involved when it comes to gene therapy, or even just talking with a genetics counselor. So if you're interested in learning more about that, just have a look at the show description. There, I have links to everything she mentioned. I hope you enjoyed this episode, and I hope you learned a little bit more about myself. I started this podcast to kind of document my journey as someone with retinitis pigmentosa, and it's kind of cool that I get to share it with you. And maybe somewhere out there, someone's getting something out of these. With that being said, the next few episodes are going to be great. We're going to cover a variety of topics, including parenting someone with a disability. So until the next episodes, wish everyone a positive, productive, yet chill week. All right, take care, everyone.